Hi, and welcome to your third podcast. One of the things you might notice about this first slide is that the learning objectives are the same as in lecture. That's because this podcast is meant to add on examples and supplement the characteristics that we use to group animal phyla, the key features that have evolved along the phylogenetic tree, and then some other structures or characteristics that we call key innovations that have allowed different clades of animal to become very diverse, or features that are just unique to individual clades. So as you go through this lecture, you'll notice that this, like in the lecture itself, you'll see the banners along the sides of the slides indicating that you're in the same topic. So the first topic that we talk about are organ systems, and those are going to be highlighted along the side in blue. In all cases, we're going to actually be looking at different characteristics, and each of the characteristics will be highlighted with a specific color. At the end of the podcast, you'll find a set of written questions that you'll need to answer and turn in at the beginning of class. You will also hear questions embedded within the podcast, so you can answer those as well. If they're not listed on the last slide, you'll have to just add them to the list of your on your paper. All right, everybody, we left off on Wednesday talking about animals and the diversity of animals. And so for your first question of the podcast, the first thing I want to ask you as you look at this phylogeny is, what's the sister group to all of these animals that you're looking at here? So your first question is, what is the sister group to all animals? When you look at this phylogeny, you should see that we have not only the groups that we talked about, so sponges, which form a monophyletic group, but we also can talk about diploblastic animals that aren't monophyletic, and then triploblastic animals that are bilaterally symmetrical, called bilaterians, and that's a monophyletic group that contains protostomes and deuterostomes. And then if we include all diploblastic and all triploblastic animals, we call those eumetazoans. And this phylogeny that you see here is the most up-to-date and recent phylogeny of animals. We left off last time talking about the phylogeny of animals, and in the other slides that I posted last time, I made sure all of the phylogenies were updated. Again, I think I told you guys in the last lecture that the major change is that the relationship between ctenophores and cnidarians changed since the last edition of the book. And so if you see any case where ctenophores and cnidarians are sister taxa, that is the old phylogeny. So when we look at this particular phylogeny, one of the things we noticed is that the nervous systems first evolved in eumetazoa. Eumetazoa is everything but sponges. So nervous systems first arose after the evolution of embryonic cell layers. So those two embryonic cell layers of the ectoderm and the endoderm. We also get around the same time the evolution of the nervous system. Most animals within eumetazoa have some sort of simple nervous system, even limited to something like a nerve net, which is just a few neurons linked together. Except for placozoans, they seem to have lost or have an overly simplified nervous system altogether. The rest of our organ systems that we have in animals evolved later, and they evolved following the evolution of the three layers of tissues, three embryonic tissue layers, the triploblastic animals. So that includes things like the digestive system, the excretory system, which controls water and salt balance, and the reproductive system, and the circulatory system. One of the features that enhanced the development of organ systems was the presence of the coelom. And remember, coelom is that word C-O-E-L-O-M, and it's that cavity that forms when the mesoderm forms in the embryo. So the coelom actually provided space for organ systems to develop. So when you start seeing things like the digestive system and the circulatory system, and even things like the excretory system. Typically, those are happening within organisms that have either a coelom or a pseudocoelom, because there's space for them to actually form. In organisms that don't have a coelom, it's not that they don't have anywhere that's specialized to do some of the functions like digestion, but they do not have the extreme organ systems. So in animals that don't have the coelom, we often find a structure called the gastrovascular cavity, and that's shown in the cnidarian. At the bottom, it's a hydra, and it's shown in the middle picture when it has tentacles. So the gastrovascular cavity is a simple opening space inside the animal that leads from the mouth, and it's used for things like digestion. So when they take up food, they do their digestion within the gastrovascular cavity. They absorb materials into the full body of the animal, but then when they produce waste, it also comes out the mouth. So in a gastrovascular cavity, you have this specialized space, but instead of having it go a single direction from mouth to anus, there's no anus, so every waste product has to come out the mouth. In organisms that do not have two openings to their digestive system, we call those animals with an incomplete gut. So if it has a single opening, it's called an incomplete gut, whereas if it has an opening of a mouth and an anus and where food travels in a single direction, we call that a complete gut. Now, in the case of the coelom, one of the other things that's advantageous is it also happens along with the evolution of muscles, and that happens because the muscles are deriving from the mesoderm. Things like the nervous system, however, were present long before the evolution of mesoderm, and so we know that the nervous system was not one of the features that actually was associated with the coelom-based body cavity. One of the patterns that we commonly see in groups of animals that have many species or that are very diverse is that they have new features or they make modifications to existing features so that the different groups often look somewhat different. In the group called mollusks, you can see pictures in this slide where there are 4 different representatives from different groups of mollusks. And you'll see that you're looking at cross sections of each of these so that you can see the internal organ systems. And while each of them has the complex organ systems we've been talking about, like the digestive system and the nervous system, they also have a few new organ systems that we haven't seen before. For example, you see that they have a circulatory system. In this case, it's what we call an open circulatory system, where the blood is not contained within vessels. But they also have other features that are unique to mollusks. So you have this pattern where you have complex organ systems and new organ systems that have evolved and are used throughout the group. But we also get new features that are unique only to the group of mollusks. Like, for example, all mollusks have a shell that is secreted by a structure called the mantle. So if you look at each of these individual panels, you can see that there is a red structure called the mantle, and that is responsible for producing any of the shells that you see. Now, normally we would say all mollusks have shells, but in the case of cephalopods on the bottom left that include things like squid and octopus, they still have a mantle, but instead of secreting a shell-like structure, they secrete something called the pen. Now, in addition to having diverse organ systems, you'll also notice that they are organized differently depending on the group of organisms. So they use the same systems, but they're using them slightly differently. And this can be important for things like acquiring new food sources or using new environments in which to compete for resources. You see the same thing within arthropods. Now, as you remember from last time, arthropods are the most diverse group of animals. They include insects, crustaceans, and then several other groups that you will learn or have seen in the diversity quiz. And again, just like with mollusks, you see complex organ systems. land insects have specialized structures called trachea that they use to breathe. This is not like our breathing system. They have direct openings to the outside of their body called spiracles. And the spiracles draw air into these hard, rigid tubes that are almost branched like our blood vessels are, and they're branched into things called tracheoles, and they reach all over the insect's body. So instead of having lungs that distribute oxygen through our blood, they actually distribute the oxygen directly through these tubes. They also, like the mollusks, have an open circulatory system, and you can see that diagram on the top left where they have a pump, a centralized pump, and they pump the blood out of what we call arteries away from their heart. But instead of it branching off into tiny little vessels, it just bathes the entire organ system, the other internal organ systems like the digestive system and their excretory system. So when you look inside something like a grasshopper, which is a land insect, it has features that other animals haven't had yet. And so we see things like the reproductive system really developed. And of course, mollusks have a reproductive system, but this just is a nicer diagram of it. You also see digestive structures, a gut, and then these things that are digestive ceca that secrete enzymes. We have vessels along the top of the body, so dorsal, that distribute blood throughout the animal. And just like with the trachea, they're not actually distributing it, and there's not a relationship between the blood and the delivery of oxygen. But as you can see, they have this open circulatory system. They have trachea and spiracles together. Some other arthropods have what we call book lungs, which are a different way to distribute oxygen that are used in aquatic arthropods. We also have a system to deal with nitrogenous waste. So nitrogenous waste is nitrogen that we have to get rid of because it's toxic. And in insects, especially land insects, they use structures called Malpighian tubules. And they're not diagrammed here, but you can see those in images. Another interesting thing that we see in animals is not just that every animal has the same organ systems, but some groups of animals also have developed unique organ systems. And so one really good example of that is in the echinoderms. They still have things like a digestive system, but they also have this system called a water vascular system. And the water vascular system is not a blood delivery system. It's not used for oxygen exchange or distributing nutrients. Instead, it's used to pump water through what we call tube feet. So in the top right picture, you can see the arm of a sea star and these little extensions coming off of it. Those are the tube feet, and most echinoderms have tube feet. If you look at the other echinoderms shown here, the sea urchin just below the picture of the tube feet, those little extensions coming off of the sea urchin are tube feet. And then in the sand dollar on the side, most of us haven't really seen a sand dollar alive. Most of us see the skeletons of them left behind, but what you see is what look fuzzy are actually the tube feet coming out of them as well. So this water vascular system has an opening called a madreporite, and that madreporite draws water in and pushes water through these canals that are shown in the top cartoon-like picture in blue. And those blue canals push water into the tube feet. So the blue structures are called radial canals. There's a ring canal at the central part. And then the tube feet are shown in blue, and they look like little eyedropper kind of structures. And when the water's pushed through them, it extends the tube feet out. So in things like the sea star, they use their tube feet to move, but they also use them to manipulate food. Other echinoderms use them for gas exchange. So sometimes you actually have not only a novel system, but different groups of organisms can modify that system in different ways. So the sea star might use it for food manipulation, whereas another uses its tube feet for getting oxygen out of water. Now, many of you may be thinking about the fact that we, of course, have vertebrates have organ systems. And in fact, this is true, But I'm not going to spend any time really talking about it because that's what Dr. Buchholz will do after spring break. He'll talk about the functions of all of these different systems as you go through the rest of the course. And he'll also, I think, so it's the nervous system. He'll talk about the excretory system, which is what controls salt and water balance, and then the digestive system, the endocrine system, circulatory system, and the reproductive system. Okay, so we just walked through the different features of the phylogeny and talked about developmental characteristics that help define our clades. But one of the other things, or one of the other patterns that we see in animals and in other organisms as well, is that when you have a very diverse group, we often see evolutionary innovations. So you can compare this to things like flowering plants, that once the flower and the fruit evolved, the group became incredibly diverse. There are similar types of traits in animals that are associated with diversification. So what we're going to do is we're going to take this pie chart, and as we've talked about before, arthropods are the most diverse group of organisms. So what are the features that they have that help them to become so diverse? And we're going to look at a few of those. But we're going to walk through some of the details of at least the first 3 of these different evolutionary innovations, some in more detail than others, to look at how those features, so the body plans, the feeding mechanisms, the paired and jointed appendages, and maybe the skeletons, how did those features change as groups became more diverse? So your second podcast question is to take this pie chart and make a list of the groups of organisms that are most diverse. So what I want is a list of 4 groups that are most diverse in the order of most diverse to least diverse. So in other words, make a list of the 4 groups of animals that are most diverse, starting with the most diverse and then going to the least diverse. Now, when we talk about these groups, what I will do is I will highlight some of the characteristics of each of these groups to get at how this feature helped diversify these groups, or how it's diversified these groups. All right, so the first thing we want to talk about is this idea of a body plan. So the body plan is really the overall structure of the organism. So when you think about something like a sponge, it has this overall structure where it's a big tube. The cells line the tube and pull water through it. So we have this normal shape of the adult and its overall features. One of the things that we see happening in many animals is a change from larvae or pupae. So these are immature forms of an animal, a larvae or a pupae, and they go through major changes in the body form. So if you take a look at the 2 images on the left, the larvae of the sponge looks like a kind of a ball, but the adult looks completely different as that long tube where we know it has the choanocytes lining it. This process where you undergo this absolute change in body plan is called metamorphosis. So if you look at the cartoon on the bottom, what we see is that we can see the early stages of the development of sponges. Porifera just is referring to the group that's the name of the sponge group. We can go from the zygote to the blastula, and then they have this stage called the amphiblastula larva, so that's just the larval stage, and it has flagella, and it's really just a sphere. As it grows into an adult, it will actually plant itself into some sort of substrate, so the bottom of the ocean, and that larva will start to grow with its flagellum up, and then it will actually take on that adult body form. Usually when we see these changes in body plan like this, there's a distinct stage that's responsible for dispersal. So in the case of the sponge, the swimming larvae is the dispersal stage, and it's going to move away from its parent or move at least somewhere where other sponges are not. And then it will have a stage where it's actually implanted into the ground. So we have a stage that's permanently attached, and we have a stage that's dispersing. It's capable of dispersal or movement. There are trade-offs to this idea. That means that it's really great to have this dispersal stage, but then there's also this limitation once they plant as an adult. And so the body plan changes can often be substantial. They can often be really great because they aid in dispersal, but then the adult stage is actually limited because it can't move away from where it landed as a. Just to give you another example of metamorphosis and how body forms can completely change, Cnidaria have two different body forms that they go through in different phases of their life. They have a medusa and a polyp. The medusa is free swimming. It's an adult. And the polyp is also an adult. But instead of it being free swimming, it's actually attached to a substrate like the sponge. So in its life cycle, and I don't expect you to know this life cycle, I just want you guys to see how the body forms change so substantially. You have a larval stage that becomes a polyp, very much like what we saw in the sponges, but it actually produces structures that are released and form this free swimming medusa. So they actually are undergoing these body form changes massively. The differences in the different forms, the medusa and the polyp, is whether they're sexual or asexual. You can see clearly from this diagram that the medusa stage of jellyfish is in fact sexual, and then the development of medusa from polyps is asexual. So not only do we have this ability to have major changes to a body plan in metamorphosis, we also have a theme that kind of works through some groups. So in mollusks, what we see is that while they have a variety of body plans, so you can see along the top pictures that, and the one on the side, these are all different forms of mollusks. These different forms of mollusks look very different from one another. And so what you see is that if you did not know, you would actually think maybe they're really different organisms. But when you really get to know mollusks, what you see is they have a generalized body plan. And so what we mean by that is the features are all the same as we go through looking at them, but they just have made changes to them, or what we call modifications. So in the case of things like the slug on the right, the banana slug, it still has a mantle, but it no longer produces a shell. Whereas the animal on the left, it has a shell, and underneath it would be the mantle and the digestive system. There are also some other structures that are really important in mollusks that are in every mollusk. So it's not just the structure, the mantle that produces the shell, it's also the radula that they use for feeding, and we'll see that again later. They also have a muscular foot. That muscular foot in the animal on the left is just used to attach and move and slide along the surface, whereas in the octopus, it's been modified into tentacles. And they also all have gills. So instead of having metamorphosis, what they have is a body plan that's generalized, and they've made changes in different groups of mollusks to that body plan. So we know that body plan we can go through metamorphosis, so we can have these major changes to body plan. We also have this idea that we have themes within a group, right? So like the mollusks that have similar features among groups, although they use them differently. We also know that there are features that evolve within the body plan that have changed the way a body plan evolves. And so one of those features is called segmentation. And segmentation refers to the repeated elements of the body. I think it's easiest to think about in terms of this millipede or centipede in the image on the left. And that animal, it just has the same repeated segments over and over and over again. But what we see throughout animals is that once segmentation evolved, different animal groups modified those segments to specialize. So the idea is that if you have many of the same element, if something goes wrong with one of them, you still have the other elements that are exact copies of it to make it work. So in something like this millipede, it has many of the same segments repeated. If one of its limbs gets injured, it still has millions of legs to deal with the walking. But other animals like arthropods and even humans and vertebrates have taken the segments that we have and did what we call specialize them. And that means change them to do specific functions. So in the case of arthropods, I'll show you this in just a minute, they have 3 distinct segments in their body. And they're actually the fusion of many segments together. And we'll look at that in the next image. In humans, we don't see our segments very well anymore, but they're very visible during development. And what's left is the happiest man on earth showing you how proud he is of his segmentation. And that's shown in his really defined abdominal muscles. So one of the things that happens with segmentation that we know has helped us in terms of the evolution of diversity is that the specialization of body regions by adding appendages allowed us to get a diversity of functions for our appendages. And that's most clear, not in vertebrates, but instead in arthropods. And I'll show you that in several. Okay, so when we look at arthropod body plans, where are the segments? So in arthropods, their overall body plan consists of 2 to 3 parts, depending on the group of arthropods that you're talking about. We know that when we look at them through development, what happens is that they are segmented as larvae, but then those segments fuse together. And we call those fused segments tagma. Okay. In the case of insects like the grasshopper at the bottom, the segments that are fused together, or the tagma, are head, thorax, and abdomen. Some groups of arthropods have fewer segments, so they've fused together more stuff, and they have a cephalothorax, which is the head and the thorax fused together, and then the abdomen. So you can see that clearly in the crayfish when compared to the grasshopper. Now, there are arthropods that have blown this out of the water and don't do any of this. So things like barnacles shown up at the top, they don't have segments at all. But the more diverse groups of arthropods all have segments. And associated with each of their segments are going to be appendages. So in the grasshopper, you see a forelimb right up at the front of the thorax. You see another secondary limb, and then a third limb associated with the abdomen. In the crayfish on the right, you see a lot more legs, and so it has jointed appendages along its entire cephalothorax and abdomen. And those jointed appendages are going to become really important because each of them, when you have many, many sets of appendages, they can be used for different jobs. And so we have this addition of segmentation. Arthropods have modified the segmentation by fusing the segments into different jobs. And to top it all off, arthropods actually go through metamorphosis as well. So they've made massive changes to their body plan in every way imaginable. There are also themes, kind of like there were in mollusks. These guys have done everything. They've taken their standard theme and modified it. They have segments and they've modified those segments, and they undergo metamorphosis. What we've been doing so far in this podcast is looking at different features that are shared across different groups of animals. In some cases, the evolution of new features, for example, organ systems, have allowed organisms to become diverse because they have new ways of doing things like sensing their environment or digesting food. There are also other features that some groups of organisms have that are unique. So things like the shell or the exoskeleton in arthropods. All of these are key features for each of these groups, but they often allow groups of organisms to become incredibly diverse. So what we're going to do when we return to lecture on Friday is we're going to look at a few other key evolutionary innovations that allowed animals to become different groups of animals to become really diverse. And since most of the time when we talk about these features or we talk about diverse groups of organisms, we really focus on things like arthropods because they're the most numerous,
mollusks because they're next, and then vertebrates. And so we'll focus on those, although I'll try to bring in different examples of unique forms of different approaches to solving problems like feeding in different groups when we go into class on Friday. So don't forget to answer the questions at the end of this podcast. If there are embedded questions in the audio, please do those as well. And I will see you guys in class.